Okay, welcome to the next lecture, everyone. If you take a look, we are already finished now with Module 3. The exam will be coming up next. Um, so, quite a quick module, but like I said, it's always better to have, I think, many smaller exams than just a few large exams. I think it's easier that way. So, let's get into the last lecture here. This one is on prokaryotic diversity. Let's talk about some characteristics of the prokaryotes. Now, we've talked about prokaryotes and we've talked about the eukaryotes, and we know that there's many differences between those two cell types. However, all cells, both prokaryotic and eukaryotic, are going to have four common structures. A plasma membrane that functions as the barrier for the cell and separates the cell from the environment, separates what's inside the cell from the outside. Cytoplasm, which is a jelly-like substance inside the cell where everything's kind of floating around. Some form of genetic material, either DNA like in our cells and most other animals, or RNA, and ribosomes. And ribosomes are the organelle where protein synthesis takes place, where messenger RNA is read and amino acids are linked together to form proteins. Okay? Prokaryotes can come in various shapes, but they mostly fall into three different categories. The cocci, which is spherical, the bacillus or bacilli, which are rod-shaped, and the spirilla or spirillum, and those are the spiral-shaped. So there's three shapes there. Here's A on the far left is the cocci. Here are the rod-shaped bacillus, bacillum, bacilli, and C is the spirillum or spirals. So they all are going to look something like that, one of those three for the most part. What I want you to remember is that prokaryotes are unicellular organisms that, of course, lack organelles surrounded by membranes. So they're not going to have a nucleus, they're not going to have mitochondria, but instead of a nucleus, they will have one single circular chromosome located in the area of the cell called the nucleoid. So it's where the genetic material is. It's a small circle, but it won't have its own nuclear membrane like a nucleus will. Most prokaryotes are also going to have a cell wall. Bacteria and archaean cell walls are quite different from each other, and they're also quite different from plant cell walls and fungi cell walls. But they are called cell walls nonetheless. The cell wall functions just the same as any other cell wall. It's a protective layer, and it's responsible for the organism's shape. Here's what a prokaryote cell might look like. Here's the cell wall. Some little pili we'll talk about. Here is the cell membrane. So the cell membrane is inside of the cell wall. The cell wall is outside. And here is like what you would call the nucleoid region, where the DNA just kind of sits. This is going to be, if it wasn't all bunched together, this would be a one circular piece of DNA. It's not held by any membrane. And then there's some ribosomes and maybe some, there's cytoplasm in there. But other than that, that's all we're talking about. Remember that I said very early on, the size difference between prokaryotes and eukaryotes is very vast. These guys are like 100 times smaller than eukaryotes. Now, this capsule is only found in some species, but it might enable organisms to attach to different surfaces and also protects it from dehydration and desiccation drying out. Some species might also have a flagellum, which helps them to move around like a spinning tail. They might have these pili, or a single, one of them is called a pilus, and that's used for attachment to surfaces and to other bacteria. Okay. Let's talk a little bit more about the cell wall. The cell wall is a protective layer that surrounds some of the prokaryotic cells, and it gives them their shape and rigidity. It's located outside the cell membrane, and it prevents them from being osmotically cut by water, or basically bursting because they have too much water inside of them. The chemical compositions of the cell walls will vary between the archaea and the bacteria, as well as in between bacterial species. Bacterial cell walls contain this molecule called peptidoglycan. It's composed of polysaccharide chains cross-linked to peptides. Bacteria are divided into two major groups, the gram-positive and the gram-negative. And that's based on their reaction to this procedure where you put some purple dye and it's called a gram stain. The different bacterial responses to this staining procedure are caused by the differences in their cell wall structure. Gram-positive organisms have a thick cell wall. It has many layers of peptidoglycan, while gram-negative bacteria are going to have a thinner cell wall composed of just a few layers of peptidoglycan and some additional structures surrounded by an outer membrane. Let's take a look at what that looks like. So here's the gram-positive with just one inner plasma membrane and then a thick wall of this peptidoglycan. That's gram-positive. Gram-negative bacteria have the inner membrane, then they have a very small amount of that peptidoglycan. You can see how it's much thinner, and then they have an outer membrane. And so that's the structural differences, but what does the difference in the gram stain look like? Well, on the same side, so on this last slide, it was gram-positive on the left, gram-negative on the right. You put this purple stain to do the gram stain, and then you wash it off, and the stain should stick to the cell based on how much peptidoglycan is present. And when there's more peptidoglycan present, more of that stain sticks, that purple stain sticks, so they stain a dark purple. When it washes away because of less peptidoglycan, it becomes pink. And so if you do a gram stain on bacteria and they turn out to be pink, they're gram-negative bacteria. If they turn out to be purple, they're gram-positive bacteria. And that's all to do with the differences in the amount of cell wall, amount of peptidoglycan they have. So that's a good test question. I could just put up a picture of some pink bacteria and say a gram stain was performed on these bacteria. Are they likely to be gram-positive or gram-negative? And you should say gram-negative. And of course, vice versa. If I put purple bacteria, they should be gram-positive bacteria. So just keep that in mind. And let me know if you have any questions about it. Let's talk about how prokaryotes reproduce. It's primarily asexual and takes place by that binary fission that we saw last time. Recall that the DNA of the prokaryote exists usually as that single circular chromosome found in the nucleoid region. Prokaryotes do not undergo mitosis, okay, so it's a little bit different. Rather, the chromosome loop is replicated and the two resulting copies attached to the plasma membrane move apart as the cell grows in a process called binary fission. The prokaryote, now enlarged, is just pinched inward at its equator, and the two resulting cells, which are exactly the same as each other, separate. Binary fission does not provide an opportunity for genetic recombination like sexual reproduction does, right? We talked about last time how that's one of the reasons why you don't look exactly the same as your brother or the kid sitting next to you or, somebody on the street. You look quite different. You have, similar features, but you look quite different. That's because of that genetic recombination. So binary fission doesn't allow that for it to happen, but prokaryotes can alter their genetic makeup in three distinct ways. The first is something called transformation, the next is called transduction, and finally conjugation. And we could talk about all of those right now. So transformation, the cell takes in DNA found in its environment that is shed by other prokaryotes that are either still alive or dead. If a non-pathogenic bacterium takes up DNA from a pathogen and incorporates that new DNA into its own chromosome, it too may become pathogenic. Of course, a pathogen is an organism that causes a disease. Transduction, bacteriophages, the viruses that infect bacteria, move DNA from one bacterium to another. Archaea have a different set of viruses that infect them and translocate genetic material from one individual to another. So here's the bacteriophage, and it comes out from this bacterium and then infects this one and gives that same transferred gene. So that's one way that it can change its genetic makeup. And then the last is conjugation. During conjugation, DNA is transferred from one prokaryote to another by means of a structure called a pilus that we saw before, and that brings the organisms into close contact with one another. The DNA transferred is usually that same small circular piece of DNA called a plasmid, but parts of the chromosome itself can also be moved. Plasmids consist of those small circular pieces of DNA outside of the main chromosome. So here you can see how this works, where this plasmid gets copied and then sent through this conjugation bridge, and now this bacteria has a plasmid. And once again, if this plasmid was for being suddenly becoming pathogenic. Well, now this bacterium that was not pathogenic is now pathogenic. Okay, so let's talk about bacterial disease. Devastating pathogen-borne diseases and plagues, both viral and bacterial in nature, have affected and continue to affect humans for our entire existence. But it's worth noting that all pathogenic prokaryotes are bacteria, and there are no known pathogenic archaea in humans or any other organisms. Pathogenic organisms evolve alongside humans. In the past, the true cause of these diseases was not understood, and some cultures thought that the diseases might have been spiritual punishment or were mistaken about material causes. But over time, people came to realize that staying apart from sick people, improving sanitation, properly disposing of corpses and personal belongings of victims that were of illness would reduce their own chances of getting sick. So let's talk about a few. The first major one is the Black Death. One of the most devastating pandemics, of course, was the Black Death, which happened between 1346 to 1361. It's believed to have been another outbreak of bubonic plague caused by the bacterium Yersinia pestis. This bacterium is carried by fleas living on black rats. The Black Death reduced the world's population from an estimated 450 million to 350 to 375 million, so 100 million deaths. Bubonic plague struck London hard again in the mid-1600s. There are still approximately 1,000 to 3,000 cases of plague globally each year. Although contracting bubonic plague before antibiotics meant almost certain death, the bacterium responds to several types of modern antibiotics, and mortality rates from plague are now very, very low. So that's one really big example of what happened before we came up with antibiotics. You went from the same basic, pretty much the same disease killing 100 million people to now killing, less than a handful of people who don't receive antibiotics. One of the scary things that we as scientists and also just the medical community have to deal with is this antibiotic resistance crisis that we are facing. One of the main reasons for resistant bacteria is the overuse and, of course, incorrect use of antibiotics, such as not completing a full course of prescribed antibiotics because you go to the doctor and you get antibiotics for, first of all, for something that maybe doesn't really need antibiotics and your body would be able to fight it off on its own, it's like a little common cold or something. But then getting those antibiotics and then taking about, 3/4 of the dose, you start to feel better and then you stop taking it. That incorrect use of an antibiotic will result in probably a natural selection of resistant forms of bacteria. The antibiotic kills most of the infected bacteria, and therefore only those resistant forms will remain. Those resistant forms will reproduce, resulting in an increase in the proportion of the resistant forms over the non-resistant ones. Another problem, like I said, is the excessive use of antibiotics in livestock. The routine use of antibiotics in animal feed promotes bacterial resistance as well. In the United States, 70% of the antibiotics produced are fed to animals that we later eat. The antibiotics are not used to prevent disease, but actually to enhance production of the products. And just so, really, I think it's important to be very cautious about using antibiotics, especially if you can prevent using them. And, if I'm not saying don't go to the doctor if you feel sick, but if it's something that is probably just a common cold, I would refrain from using antibiotics until you really do need it, because once you start to become resistant to them, or you get infected with resistant bacteria, things go downhill very quickly after that. Here's an example. I'm sure we've all heard something about MRSA. This is methicillin-resistant Staphylococcus aureus. So coccus, remember what that means, they're circular. Staph aureus causes staph infections, and methicillin is a really strong antibiotic, but methicillin-resistant Staphylococcus aureus they do not respond to methicillin. And you can see that right around 1993, the year that I was born, there was only about 1,900 cases to now 2005, and you can imagine that this has just continued upward on this trend to 2020. there were 368,000 cases of methicillin-resistant infections. And so it's really unfortunate because the amount of methicillin resistance and the ways that antibiotics are able to reproduce so fast, the resistance can evolve within bacteria much, much quicker than we can come up with new strains of antibiotics. And so this is why it's really important that just make sure that, well, when you go to a doctor, it's for a good reason. When you go on antibiotics, it's for a good reason. And things like that. So just overall, keeping in mind what happens from the poor use of antibiotics. But let's again, I always try not to leave you on a scary note. Let's talk about the beneficial prokaryotes. Not all prokaryotes are pathogenic. In fact, pathogens represent a very, very, very small percentage of the diversity of the microbial world. Our life on this planet would not be possible without prokaryotes, right? The first photosynthetic organisms were these bacteria, basically blue-green. They used to call them blue-green algae, but they're actually bacteria that can do photosynthesis. Without that, we don't have oxygen in our atmosphere, and of course, we know why we need oxygen. Before I get into anything else, we've talked about some other things like prokaryotes are useful for. One thing that I always like to say, and I think I might have already said this in this course, is if I was to put you in a blender, a hypothetical blender, and make a human smoothie, and then I had a cell counter, you'd be less than 50% human. The rest of you would be prokaryotic bacteria that lives on you all the time and actually keeps you healthy. So that's really important. You have mutualistic, for sure, mutualistic bacteria. One example is that very sick people actually end up taking, for lack of a better term at the moment, poop pills. And basically they're pills of actual freeze-dried poop in a way to give you good, healthy gut bacteria for people that have been very sick, who have been on antibiotics, that have killed their gut biota. That gut biota is very, very important for digestion and getting these nutrients that we need out of food. And so you can actually give good cultured bacteria back to people so that it can reline their gut. And that mutualistic relationship, they get a place to live, get some nutrients, and you get the benefits from staying healthy and getting those bad bacteria out of your system. If they're living in that place, the bad bacteria cannot live there. So at the very least, there are definitely mutualistic relationships, if not commensalistic relationships. A commensalism is a theorized interaction where one organism gains a benefit and the other organism is neither positively or negatively affected. I tend to argue that this is impossible. Even if it's very, very slight, there's going to be some positive or some negative for the other organism. Right, we'll talk about, when we talk about organismal interactions, which is one of my favorite topics, we'll talk about commensalisms, where scientists used to believe that an interaction was a commensalistic relationship, again, where there's one positive and one neither positive or negative interaction, but then as we continued to look at it, we actually found that that wasn't the case. But when it comes to this gut bacteria, you could argue in some instances that they don't really provide you with anything and they get all the benefit. But really, I think that they do provide you the benefit, which is they are not infecting you, and another organism, another prokaryotic organism, could come in there and certainly harm you. Another example of a beneficial prokaryote is the ability to do bioremediation. One example is when there's oil spills, bacteria that break down that oil can be released. And that's called bioremediation, where bacteria make a toxic compound safe for the environment. And so I just hope that leaves you on a good note, thinking about these bacteria and also archaea. Remember, there's no archaea that can affect animals or humans. And so just interesting to think about. Let me know if you've got any questions on this, guys. Congratulations on completing Module 3. Get ready for the exam, and I'll see you in the next video.